So I'm gonna try to answer this very general question, and Oded actually mentioned that it's the question that he asked. And I think anyone who's talking today could have entitled his presentation with this title. Yet this presentation is going to be very different from what you will hear otherwise, simply because I don't have any data to show. I'm just investigating this question, like, can in principle non-genetic inheritance be important for evolution with models? And to answer such a general question, we need to make models that are very simple and general. And it's quite unusual for me, so I'm just going to show you what I usually do and how it's different from that. So what we do in the group is generally study the evolution of regulatory networks. And for example, gene networks where you have gene products that regulate each other's expression. And we use that to study general questions about evolution, like the question of the evolution of multicellularity, for instance. Because in multicellularity, you have a single genome that's able to generate different cell types. And exploiting noise in gene expression could be a way to do that. So we want to, we are building models of gene networks with noise in gene expression to see if this could be a trigger in the early stage of the evolution of multicellularity. And the other type of networks that we study are endocrine networks, where you have interactions between hormones and receptors, and this interaction governs the allocation of resources to different structures. And with this type of models, we study how, so we have dynamics of binding between hormones and receptors. They govern the intake of nutrients by different structures. And with that, we generate combinations of traits, like fecundity, survival, for instance. And with a given genotype, you can reach a given combination of traits. And of course, with genetic variation in the hormones and receptors, you can get phenotypic relationship, like genetic variation that establishes in the population. And actually, with this kind of model, we study the evolution of the shape of this relationship, how it can evolve through time and in different conditions. So that's what we do, and you see that there is some physical interaction involved and stuff like that. And these are complex models. And what I'm going to do today is show you a very different and much more simple model than that. To study the contribution of non-genetic inheritance to evolution. So I used a trait that's a beak. I don't know if you can see that. So we actually don't have a specific trait in mind, but this is just to illustrate what I'm going to say. So it's a beak size, which is the very famous trait in evolution. And of course, there's an underlying cellular structure to this trait, and you can have cellular features that translate into changes in this trait, and you can have developmental dynamics that also change the trait in question. And well, we are all here to talk about epigenetic inheritance today, so I won't have to convince you too much that these features can be affected by DNA changes, of course, but also by non-genetic changes that may affect the trait. And this could be methylations, this could be changes in RNA concentration and stuff like that can be transmitted from the parents to the offspring, so that are heritable. And of course, the trait is subject to selection. I will consider stabilizing selection in this talk. So you have a specific trait value that's optimal like here, to the environment, for instance, to the size of the seeds in the environment. And if you change the environment, if you change seed size, then you will have a different optimum for the trait. And of course, if you have such an environmental change, and assuming that you have heritable variation, you should see a change in phenotype through time. And of course, the question we're asking today is whether this change occurs through change in DNA, so the genetic medium of inheritance, or does it occur through change in the non-genetic medium of inheritance. And so I'm a theoretician, so I need to find a feature that changes between these two media. And one obvious one, it's been mentioned today, is the frequency of changes. The non-genetic medium typically changes much more frequently than the genetic medium. And this could be the end of my talk. Like, it changes much more frequently, so you don't have to wait for a long time before the first epi mutation, if you want. So it should be the one that governs adaptation, that occurs faster, right? And when we do this reasoning, and it's something that we often read, actually, we make the implicit assumption that the epi mutations and mutations have similar effects, or like effects of similar amplitude, I guess. And this doesn't have to be the case, and this is the question I'm going to ask. Is it possible to have a medium that mutates very frequently and yet has strong mutational effects? Okay? So that's the question I ask, and I ask this question and try to answer these questions with models. So the model I present today is extremely simple. We have an epigenetic medium that can be in two states, 0 or 1. We have a genetic medium that can be in two states, 0 or 1, and you can switch between these states. The mutation rate, if we can, or epimutation rate, of the non-genetic state is mu E, and the mutation rate of the genetic states is mu G. And of course, mu E is larger or equal to mu G. And the two traits, the two states, the two media contribute to a trait that's called phi I. So I is just for individual I here. And the contribution of each medium to the traits is dependent on another locus, that changes the value of alpha I. And alpha I is the relative contribution of the epigenetic medium to the trait. So the trait value is the weighted mean of the genetic and epigenetic medium, and it's weighted by the contribution alpha I. And alpha I is supposed to be genetically encoded, and so it changes with a mutation rate mu G. So if you have a large value of alpha I, a large contribution of the epigenetic medium, the trait will be much more dependent on the epigenetic state. Okay. And then we have recombination. It's possible to have recombination between these two loci. The recombination rate is R. So if R is 0, they are linked. And if R is 0.5, they are independent. And fitness is calculated as a function of the difference between an optimum, that's represented here, and the phenotype. So if the phenotype is very different from the optimum, fitness will be low. And this optimum can change in time. Oops, sorry. It can change in time. It can take two states, 0 or 1, and it can change with a probability PC. So PC can be 0 and the environment is stable, or PC can be positive and then the environmental changes at some frequency. Okay, and here are the first results. So we are in a stable environment, PC equals 0. And this is one simulation over time of the non-genetic contribution, the mean in the simulation, non-genetic contribution. And we see in this simulation that the non-genetic contribution has increased through time and reached this value. And this simulation is when the mutation and epimutation rates are equal. So it's kind of a control, I would say. So if alpha is here, if we have this value of alpha, we have a dominant contribution of the non-genetic medium. And in this, when alpha equals 0, we have a dominant contribution to the phenotype of the genetic medium. And in this case, we see that we evolve a large contribution of the non-genetic medium. And if we do more simulations, which we see that it's a bit random where we go, we can have large contributions of the non-genetic medium or or large contributions to the, of the genetic medium. And on average, we expect 0.5, which makes sense because the two media are identical. They don't differ. So there's no reason that one should be favored over the other. Now, if we increase the epimutation rate to 10 to the minus 3, so it's 1,000 times more than the previous one, we see that the evolutionary dynamics are very different. We see that the majority of simulations lead to the evolution of a very small value of alpha, so a very small contribution of the non-genetic medium. Okay. And it's in the vast majority of simulations. And we can do that by varying the non-genetic mutation rate. And we see that at some point when the non-genetic mutation rate becomes high, there is selection for decreased contribution of this medium, which makes a lot of sense actually, because if you have a medium that moves the phenotypes away from an optimum, and this optimum never changes, you shouldn't favor a medium that moves this optimum. And so you should favor the larger contribution of the less mutable medium. And what I want to say here, you shouldn't take these values for granted. For instance, what happens when there is not enough selection acting on the contribution will depend on details that that have been fixed arbitrarily in this model. For instance, the neutral expectation of alpha will probably depend on mutation bias acting there, and we assume no mutation bias, so it can increase or decrease with the same probability. But probably in nature there are some mutation bias. And also the non-genetic mutation rate at which the switch occurs is dependent on many things. It depends on how much selection there is against deleterious mutations. So we have two parameters for that. We have parameter S that I mentioned before, which is the link between the phenotype and fitness. And so we can change that and move this curve. And also we can improve selection by changing the population size. Oops. And this is what we've done here. So we have increases population size when we go there. And we see that in larger populations, the switch occurs at lower non-genetic mutation rates. So, I mean, if we want to make real predictions for a real system, we'll have to get assumptions about this system. This is very general work, and we shouldn't say that, oh, when we are at 10 to the minus 3, we switch toward a lower contribution. It's not how this works. Okay. So the take-home message at this point is that selection will act against a large contribution of non-genetic media in a stable environment, simply because having variation when the environment is stable is deleterious. Okay. So it's a bit obvious. And now we will switch to the situation where we have environmental variation. In which case, we can suppose that some contribution of non-genetic media might sometimes be favored. So this is the curve that we've already seen in a stable environment. And when I increase the probability of a change to 10 to the minus 3, here's what happens. So we see that the large contribution of the non-genetic medium can be favored for a large range of non-genetic mutation rates. Okay, probably because they allow the organism to follow the moving environmental optimum, and it decreases back when the non-genetic mutation rate becomes high. Simply because if you have a probability of change of 10 to the minus 3, it means that on average the environment changes every 1,000 generations. And so, okay, there's the time when it changes and it's good to have variation, but then there's the long environmental stasis where having a non-genetic mutation rate of 0.1 is highly deleterious. Okay, so yeah, that's it. And we can, this is exactly the same results but presented in a different manner. So we have the non-genetic mutation rate here. We have the probability of environmental change here. And we see that when the PC equals 10 to the minus 3, we have a large range of non-genetic mutation rates where the contribution of non-genetic medium is highly favored. And we can represent that for all other probabilities of environmental changes. And we see that as the probability of environmental changes increases, we have an increase of the non-genetic mutation rates that has the highest contribution. So we see that the optimum in the non-genetic in alpha, the mean expected non-genetic contribution increases as the probability of environmental change increases. And this is very this is a result that is very similar to some results that can be obtained in bit hedging theory, for instance, which consider typically very high switching rates between phenotypes, but it's the same kind of results and explanation. And now we can play a little game. Can you tell me the major difference between these two graphs? Anyone? Do you see it? What?
The white at the bottom and the right.
This one?
No, a row above.
The
Another one above.
above, sorry. This one?
No, one more. Yeah.
This one. Oh, okay, there's one. Yeah, that's true. Well, it's
not
it's still a very small difference, actually. And actually, the difference between these two graphs, you can't see it, and I will show you now. The difference between these two graphs is that in this situation, the recombination rate is 0. In this situation, the recombination rate is 0.5. And it makes almost no difference except for this one and this, actually. There's a small difference here, too. So recombination is between the locus that contributes to, generate the value of alpha, the contribution, and the locus that contributes to the trait, like the epilocus and locus. Okay. And that's very, we were very surprised because we expected a large difference between these two situations and in between, a large effect of the recombination rate, simply because we thought of the contribution as something a bit similar to the evolution of the mutation rate, right? It's kind of the same type of questions. What's the optimal mutation rate in that situation? And for the mutation rate, it doesn't work this way. We would have a major difference between the two. Like the mutation rate would evolve when the recombination rate is 0, so when the modifier and the effective locus are linked. But it wouldn't be the case when there is no linkage, simply because the mutation rate relies on, takes advantage of the mutations that it will generate in the future. So we need to have linkage because otherwise you have a large mutation rate. Offspring have mutations that can be adaptive. But then if you don't have linkage, you lose this advantage. The modifier locus loses this advantage. And so what it says is simply that the modifier locus of alpha doesn't rely so much on future variation that can be generated in the offspring, but it already exploits the existing variation in the population. Okay. And so the take-home message at this stage is that traits under fluctuating selection may rely much more on non-genetic inheritance than traits in a stable environment. And actually, if you have like a variety of traits for an organism, we can predict that the contribution of one specific medium, depending on its mutation rate, will depend on the amount of environmental changes that affect the trait. Okay. And now, what we want to do next with that, there is a question. So what we've done so far is fixing the mutation, epimutation rate, and see how the mutational effects evolve. And this is quite a restricted setting because we know that mutation rates can also evolve. And so what we want to do is to do both, like joint evolution of mutation rates and mutational effects. And this is, I think it's a very interesting question because if you can let both evolve, if you can have the evolution of mutation rates and mutational effect, in a given setting, like a given frequency of environmental changes, for instance, I think what will evolve will be only one combination of mutational effect and mutation rate. So you will have a mutation rate that evolves and that corresponds to the rate of environmental change and a large contribution of this medium. And now the question we can ask with this type of model is, why do we have a diversification of inheritance media? Why do we have several inheritance media and not just one? And probably the answer is due to the fact that different traits don't have the same, are not subject to the same type of selection. And you have traits that are subject to frequent changes in the environment and traits that are very stable in the long term. And so that's probably a good reason to have different mechanisms of inheritance. And I have a different thing that I want to talk about today, and that's something I'm thinking about, but I wanted to share with you, but I'm not sure about this reasoning, actually. So the assumption that I've made is that we have two media that can contribute in the same manner to a given trait, okay? But then these different media act differently on gene expression, for instance. If you take gene methylation or most non-genetic inheritance mechanism, what they do is often they change the expression of a gene, right? So they have this kind of restricted mutational space where they can do only this thing. And it can be very important, of course, but it's more restricted than DNA, for instance, DNA changes and mutations that can change expression, of course, but also the protein sequence, for instance. And so there's a change that this restricted mutational space leads to restricted possibilities of changes of the phenotype. And the way I represent that in my mind is that we have different phenotypes represented by these colors here. And we have mutations of a given medium that go from one phenotype to another one, all right? And I think it would be useful to try to understand how these connections evolve. Like you can suppress some, you can make some new, and see how this space evolves, right? For different media. And let's consider that we have recurrent changes in the environment that favor this phenotype or this one. Okay, these two phenotypes. And let's see what the different media would do. The first media, the genetic medium, has small mutation rates. And so the population may be at this stage, and sometimes some mutants may go there, here. Okay? And since it doesn't occur very often, the model I showed you today says that these connections won't be lost, like it will probably evolve neutrally. It's not enough selection pressure to suppress these links, right? Whereas if you have a medium that mutates very frequently, well, then if you're here, if the population is here, and you have mutations going there, like 10th of your population, of your offspring go to this phenotype, well, it's obviously very deleterious, and you should, if it's possible, you should suppress this link, right? And in the end, what you would end up with is something like that. So you have a phenotype space when the mutation rate is very high. The phenotype space that's accessible to this medium should be very restricted, right? So, and the thing is, this medium should then contribute a lot to this kind of adaptation, going from there to there, to there, to there, etc. But then, what about a novel niche, like something that would be here? Imagine that there is a new phenotype, like going out of the water or something like that, an evolutionary innovation. Then this medium, the pink one, the non-genetic one, would be completely unable to reach this novel phenotype, whereas the genetic medium, the more stable one, would still be able to have in a few mutations or maybe many mutations, would still be able to generate this innovation. And so I think that the contribution of non-genetic media in the short term, like fast adaptation and usual adaptations, is probably important to answer the first question. But I think for the long-term innovation or evolution, I think by definition the contribution of non-genetic media should be very restricted for that. And that's the last slide of my talk. So this work was made with Sylvain Charlat, who is also a researcher at LBBE. We have a preprint if you want to look at it. And actually it emerged during discussions in a discussion group on the genotype-phenotype map at the LBBE. So the group is now dormant or extinct, I don't know. But it was very interesting. We had several meetings and it was great. And this is how we ended up working on that, so it's cool. And last one is some advertising about the next conference that we organize in Lyon, which is Mathematical Models in Ecology and Evolution. So it's a very cool conference on modeling, and if you want to come, or well, you're welcome to. And until the 1st of December, we accept proposal for mini symposia, so you should think about it. Thank you for your attention.
Now, I was wondering, in your model, the difference between the genetic and non-genetic parameter is just the frequency of genes. But we could think that there could be another parameter that would be for how long the phenotypic impact would last in the two cases, because you go for 10 to the 5 generations, but we could imagine that the non-genetic would dilute, or I mean, it would faint more rapidly than a genetic change. I mean, the effect on the phenotype. I don't know if I'm clear.
Sure. So yeah, I understand. Actually, it's already in the model, in that a smaller mutation rate would make the phenotype stable for more generations, right? If you have a mutation rate of 10 to the minus 1, every 10 generations on average, you would switch back to the ancient phenotype. And if it's 10 to the minus 2, on average, every 100 generations, you will change. But now, what's not in the model, and probably that's what you had in mind, is a bias in the mutations. Like you could have switched from 0 to 1, for instance, that are easy, very frequent. When you're at 0, it's easy to go to 1, and then you stay at 1 for a long time, which would be mutation bias, and it's not in the model, and it could be in the model, of course. We could do that very easily. I don't know what would change.
Yeah, back to the notion that epimutations mostly affect gene expression as opposed to gene products. At the same time, these epimutations could have sort of a very strong effect on gene expression, whereas for point mutations, it's much rarer for them to do affect gene expression. So my question to you is, do we have an indication that the first steps in evolution are more towards making new gene products or playing with gene expression levels or patterns? And basically, could we think that epimutations have a more important role to play in early stages of evolution as opposed to, I don't know. Does it make sense what I'm saying? Maybe not. Well, it makes sense according to.
Okay. I'm not sure. What's the first stage in evolution? Is it early on the phylogenetic tree? Okay. Yeah, sure. I think there are some papers showing that changes in gene expression are very often involved in adaptation. So sure, yeah, I would agree with you. And probably this is a picture that's qualitative. If we wanted to make quantities, of course, DNA changes affect gene expression less often than non-genetic changes. So probably it would be like a huge specialization of non-genetic media to changes in gene expression, and some specialization of DNA changes to changes in gene products. And so they have different spaces, of course, and the question is, why do they have different species? Why does it matter? So yeah, something that we will do in the next few years, I guess.
Do you think in your model it would be interesting to include a correlation between the mutation rates and the environmental changes?
Okay, like a response to stress, for instance. Yeah, of course, it's interesting to do that. Yes.